Hey everybody, this is pretty strange, but here we are, so let's do it. So let's talk a little bit about this equilibrium lab. I have posted a video that Scott kindly made, kind of walking through the experimental setup. You guys have already done essentially all of this, so all the techniques we use in this lab, we used in the kinetics lab that you guys just completed. So that involves making standards, making solutions of accurately known concentrations, right, using volumetric glassware, so volumetric pipettes, volumetric flasks. And in week one, essentially the idea is we want to make a group of standards, so solutions that we accurately know the concentration of, and we want to analyze these standards using our Spec 20s, measuring absorbance values. Okay, so the reaction we're going to be studying over the next couple weeks is a reaction between iron 3 and thiocyanate. So these react to form a complex, which is an iron 3 thiocyanate complex. And what we really want to study is we want to study the equilibrium constant of this reaction. Okay, so how are we going to do this? Well, in week one, what we're going to do is, one thing you will notice when you look at the video, the iron 3 solution is approximately colorless. Maybe it's a little orange color, depending on the concentration. This is certainly clear. When we mix these and make this iron thiocyanate complex, you'll notice that this is some kind of orange, maybe brown-ish color. Okay, so what does that tell us? That tells us if we want to look at the absorbance of visible light, not much, not much. This is the thing that's going to be absorbing visible light. And we can determine the concentration of this complex essentially by measuring the absorbance of visible light at a particular wavelength. So remember Beer's law, which tells us absorbance is directly proportional to the concentration of the absorbing species. So in this lab, this 2-week lab, we're going to be measuring absorbance values at a particular wavelength and relating those absorbance values to the concentration of this iron thiocyanate complex. Okay, remember absorbance does not equal concentration, but it's directly proportional to. Okay, so the whole goal of the first week's experiment is to figure out the relationship between an absorbance value we're going to measure and the concentration of this product. So to do that, we're going to make standards and we're going to construct a calibration curve exactly like we did in the kinetics experiment. So, part one is we're going to make a bunch of standards that we accurately know the concentration of this complex. We're going to measure absorbance values and we're going to plot a calibration curve. Okay. How do we know the concentration of our iron thiocyanate in our various standard solutions? Okay, well, page 65 in your lab report. Let's go through this and let's talk about how to find the concentrations of this iron thiocyanate complex. Okay, so what we're going to do is we're going to take stock solutions. So bear with me here for a second. So we have a stock solution of 4.0 times 10 to the minus 2 molar iron. We have a stock solution of the same, 4 times 10 to the minus 4th molar SCN minus. Okay, so notice in this particular experiment, week one, we have a much higher concentration of iron than we do thiocyanate. Okay, the reason being we'll see in a minute. What we need to do is mix a series of standards from our stock solutions, and we're going to do this volumetrically. We're going to do this in volumetric flasks. Remember, using our glass transfer pipettes. Okay, so let's just go through, let's do flask number 3. Why not? Okay, so flask number 3, what are we doing here? We take 5 mils of our iron with a transfer pipette. We take 2 mils of our thiocyanate, and we mix them into a 10 ml volumetric flask. Okay, so what we want to figure out is what are our initial concentrations of iron in this flask and thiocyanate in this flask. Well, remember, it's a simple dilution calculation. M1V1 equals M2V2. All right, so let's do iron. How do we find the concentration of iron in this flask? Well, our initial molarity was 4 times 10 to the minus 2. We are taking 5 mils of that. We are diluting to a final volume of 10 mils. So we can figure out what our final molarity is. So what we're essentially doing is diluting this by a factor of 2. So our final molarity will be 2 times 10 to the minus 2 molar iron 3 plus. Okay, so that's the initial concentration of iron in this standard we just made. Could we do the same thing to find the initial concentration of SCN minus in this flask? Yeah, same idea. M1V1 equals M2V2. So we will find that in this flask, our initial concentration of iron, which we just did, so that little knot means initial, is, what do we say? Diluted by a factor of 2. So 2 times 10 to the minus 2 molar. We can find our SCN minus initial, just M1 V1 equals M2 times V2, is 8 times 10 to the minus 5th molar. Okay, so the point of having our iron much more concentrated than our SCN minus is we are essentially forcing this reaction to completion. So just real quick, this is an equilibrium, but we know a couple things about this reaction. The equilibrium constant is pretty large, which means products are favored. Also, by adding a large excess of iron, think about Le Chatelier's principle, by adding an excess amount of iron, a large excess of iron, we are driving that reaction far to the right. So under these conditions, it is going to be a good assumption to assume this reaction goes 100% to completion. Again, the reason being is we've added so much excess iron, shifting the reaction towards products. Okay, so what can we use this to figure out? Well, this now essentially becomes a limiting reactant problem, where our SCN minus is much smaller concentration than our iron. So we can assume the following, that all of the SCN minus reacts. All of the SCN minus reacts to produce our product. So what can we say in this assumption, assuming 100% of the limiting reactant reacts? Well, 100% of our SCN minus will react, so the concentration of this iron thiocyanate complex should be the same as the concentration of the SCN minus. So in this flask we just made, flask number 3, the concentration of our FeSCN2 plus will equal the concentration of the SCN minus. And that's going to be true for all of the standards we do. So we should know the concentration of our iron thiocyanate complex. It will be the same as the SCN minus concentration because this is our limiting reactant and we're forcing the reaction 100% to completion. Okay, so at this point we have made all of our standards and we know the concentration of our iron thiocyanate complex in each of our standards. So now what we need to do is measure absorbance values. So from the video that Scott recorded that I posted for you, you should be measuring absorbance values for all of our different standards that we know the concentration of. So you should compile data where we know the concentrations of this complex, We measure absorbance values. Now we can figure out the relationship between absorbance and concentration. Okay, so how do we do that? Everyone on board? Any questions? That's a joke. All right. To find the relationship between absorbance and concentration, we are going to make a calibration curve. We should have, I think, how many? We got 5, but the first one doesn't have any product. So really we're going to have essentially 4 data points. We can connect these with a straight line. Microsoft Excel, as you're well versed in, will give you an equation for this straight line. Now we have the relationship between absorbance, our Y value, and concentration of this iron thiocyanate complex, which is our X value. Okay, what can we use this calibration curve to do? Well, now let's say we had an unknown solution that we did not know this concentration of. Well, if you were simply to measure an absorbance value, you could figure out that unknown concentration by solving for X, just like we did in our kinetics experiment. Okay, so we're going to construct this calibration curve and we're going to use it for a couple things. In week 1, we not only make this calibration curve, but we also have to figure out the unknown concentration of some solution that you don't know the concentration of. You can measure an absorbance, use this equation, and figure out its unknown concentration. From the unknown concentration of this complex, what you need to figure out is an unknown concentration of SCN minus, and that is essentially the unknown that you're starting with. Okay, so just work through the lab report, it should make sense. Cool, so week 1 is twofold. Construct your calibration curve, figure out the concentration of some unknown solution. All right, what we will then do in week number 2 actually, I'll just do a different video. All right, that's week 1. Again, use this video along with a video that Scott recorded showing you how to take measurements and showing you what the actual data is going to be. So use, for all of your lab report, all your calculations, use the data that Scott measures in that video. If you have any questions, just let me know.